Hello, welcome everyone, in person and also online, to the Evaluating Multi-Omics Data Standard Integration Challenges at Building Solutions session. So this session was organized by the Multi-Omics Metadata Standards Integration, the MOMSI working group. And thanks a lot for being with us today. So just as a reminder, for new members and also old ones, these are the 6 guiding principles of the RDA community. So before we get into the session, some logistics and some housekeeping notes. So these links on the working group information, on our new space in the RDA website, the proposal for this session, as well as the shared Google folder that we have, where we have all the working group documentation. It can be found on the collaborative notes, and also I will share the links in the chat for the online participants. An important note is that these documents, the shared documents that the working group members are collaboratively working on, they're closed and editable for members only. So if you're not a member of the group and you would like to continue, please join. We'll share the link shortly. How to participate, please sign in and take notes in the collaborative notes document. This is the QR code for the people that are in the room and also online. I've shared it in the chat. I will keep sharing this for the people that join us. The session, as you heard, is being recorded, and the organizers will make the recording publicly available. If you would like to speak, those in the room just do this. And those of you who are online, please raise your hand and unmute, or feel free to add to the chat. And finally, again, please take a moment to read about the RDA guiding principles as well as the code of conduct. These links are also available in the collaborative notes. So again, how you can join us, please scan this QR code if you're not already a member of the working group, and make sure that you sign up, you join the meetings to stay updated with all the updates and developments of the group. So some introductions. I'll start with myself. My name is Athena. I work for RDA Europe and the RDA Tiger project, and I'm the working group facilitator. In the room with us, we have Lindsay Anderson, staff scientist at the Pacific Northwest National Laboratory, and also a fair sharing omics champion, as is a working group co-chair. David Mollick, a computational biologist at the United States Department of Agriculture, couldn't be with us today, but he's also a co-chair of the working group. And with us also online is Tim van der Bos, bioinformatics researcher at Ghent University, and he will be also speaking later on. So just a few things about the agenda. Welcome introductions. We already took care of that part. And I'm going to give you also a refresher on the statement of work of the working group shortly. I'm going to pass the floor on to Lindsay, who's going to give us an update of the already completed deliverables and also current developments of the ones that are in progress. Hopefully we're going to engage in a discussion, so we invite you to participate in that. And then moving on, we're going to have a flash talk presentations round under the theme of multi-omics data standard integration challenges, gaps, and community use case developments. So here we're going to hear from Tim, Bert, who's also online, and also Catherine. And finally, we're going to close off by some final remarks and next steps. So, okay. And as a refresher, if you're not already a long-standing member of the group, and also for the old members of the group, the MOMSI working group aims to address the many challenges in evaluating both the accuracy and the completeness of existing omics community standards. So the working group ultimately aims to provide clear recommendations that are based on careful curation and dissemination of review deliverables, all of this in support of the state-of-the-art developments in the multi-omics data standards integration. So this was my sort of brief introduction, and welcome to the session. I would like to pass the floor on to Lindsay to give us an overview. Oh, sorry, of course. And yeah, the floor is yours.
There's multiple things. Okay, perfect. Okay. Mostly, no, I'm okay. Go ahead. No worries. For those who are less familiar with omics, since our landscape review, which defines a lot of the terminologies and the things I'll be talking about, are closed unless you've already joined. Just to refresh about what omics is, and of course it would fit only with this working group to use standardized terminologies and ontologies for some of these. So omics is the collective characterization and quantification of pools of biological molecules that translate into structure, function, and dynamics of organisms and an organism or organisms. So, and the multi-omics definition is by our own group, and that merges multiple omics data types, leveraging high throughput technologies and methods for quantification and characterization of large complex pools. And the integration and analysis provides a closer look into the interactions at the molecular level, and then also improving our understanding of the biological and environmental dynamics of living organisms. Okay, now I can put my phone down, sorry. Just in case, because, you know, omics technologies are comprised of a lot of different instrumentation, and really this working group is very focused on what's almost subject agnostic, even though there is a lot of subject matter investigations in these areas. So here's some omics technologies that we're focusing on. So specifically sequencing and mass spectrometry as the core technologies, and then genomics, proteomics, and metabolomics as our core parent domain spaces. So you're either very domain heavy expert in one or another. And then there's these subclasses, right? You have epigenomics, transcriptomics, metaproteomics, and lipidomics. So our 4 deliverables are very scoped in sequence, and it's very centered around illuminating what exists, not reinventing the wheel, and drawing attention to all the standard work that's been done in this community. So genomics, proteomics, metabolomics has been establishing standards for over 20 years each. So that is a lot of defined standards in these different areas. So the ones I'll discuss today are our landscape review, and then how does that landscape review lead into our omics collection at fair sharing, and I'll also describe why we're using that. Okay, perfect. So those who aren't familiar with fair sharing, I encourage you to scan this QR code to learn a little bit more about the standards we're focusing on. These specific 4 standards, reporting guidelines, models and formats, terminology artifacts, and identifier schemas, are the core standard elements that really build FAIR into the domain space of multi-omics. So if you're not familiar with what each one of these would look like, we have an example in mass spectrometry here and sequencing. So we have MAPE guidelines and MIXS guidelines, which some of you might be familiar with. We have formats like MZML or a FASTA file. These are typical data file formats. The PSI MSCV is a controlled ontology specifically used by the proteomics community. It is well defined. It is well used in all systems and databases. EFO is an adoptable ontology that you'll find within the MIXS actually. So there's a lot of interrelation here. And so we have also the USI identifier, and this is supposed to be a bioproject identifier. So these are things we see all the time. So these were the types of things we were working on in our landscape review. Okay, so this is, I apologize if this is poor. So the goal of the initial deliverable for the landscape review is everyone who joined is a domain expert in some area. And if you're not, this was a great place to learn. We split out folks into teams within genomics, proteomics, and metabolomics. And we outlined our curation sheets to conform to the types of standards that we were collecting, which are those main 4. And then we had certain criteria involved with it too. So we wanted to know, are they established? Are they established and maintained? Is development uncertain? And so this helps us also identify what is the state of what exists out there. So all the genomics experts in our first couple working groups populated all the standards they knew about, what they use, what they're aware of, what they've developed. And the second or the third column, we have the country. So this also helps illuminate, are these globally represented? Are these specific to country or maybe specific to certain communities? And then we have the domain and class and subclass. So if by row, this particular terminology was specific to only genomics, or maybe it was genomics, transcriptomics. So if you join the working group, you'll have access to this and you can browse it. We have a full data dictionary in there, affiliations tab, all the good stuff. So this was a great way to include everyone in this curation activity, but also format it in a way that will help lead us into our next deliverable, which is the collection. So it's really important, I think, up front when you're doing work within a high multidisciplinary group, that you think ahead and how you're going to maintain and sustain that work. So all this work that we did in this Google Sheet will eventually be transformed into something that's more machine actionable and also sustainable. I'll touch on that later. So this is just a good example of also, these are the types of things we'll define in the landscape review. So, you know, if you're unfamiliar with what the definition of these different standards are, or maybe it's unclear for you about what encompasses the classes or subclasses that we're focusing on for here. And so this is something that's included that you can kind of browse through. This is just a quick capture. So these are, of course, our core domains, but one thing we're also pulling in here is universal. So universal is defined as something that helps support the FAIR behind the domain standards. These will be things that you're very common with, like DOIs, maybe the ISO date and time standard. Maybe it could be ISATAB schema, things that help build more actionability around domain standards and help supports integration. So this is a really great spread. You can see within these different domains, which ones have, you know, the largest collection of terminologies, formats, guidelines, schemas, and the multi-standard applicable means that maybe they both have adopted it. So you could use it in genomics or proteomics. They might be actively using it in both already. So this is kind of helping us capture what's out there. Other community driven, that's also going to be very FAIR specific. So that's the things like the FAIR principles or maybe the CARE principles, something to be noted, but may not be specifically relevant to a domain, but something you want to also include. So something really unique about our working group that we did in this landscape review is if you hear about the data lifecycle all the time, no one really provides great high level detailed examples. We use the RDM kit as a baseline. I think many people are familiar with it. I think this can apply across any data lifecycle that anyone's really adopting, honestly. But we specifically drilled into certain areas within this. So it was the plan, process, I think collect and analyze and share components, reuse. So how do these 4 standards fit within each block of these elements within the data lifecycle? So here you have experimental designs, sample preparation, data acquisition, what instruments are you using and what data type is it? Feature detection. So are you mapping identifiers in this particular part of the landscape lifecycle? So what we wanted to do is think about things in a way that helps support future planning of standard selections, but also giving expert level knowledge to non-experts by sitting together as experts within a room across these domains and defining them as best we can, of course, right? So a good example in our sheet is we have these, I think, oh, perfect. The plan, collect, process, analyze, preserve, sharing, reuse. There was a request, we're still making sure to iron out some of these definitions. So across each standard, you know, it may apply something, maybe some standard, let's say like Go ontology, right? It might help you more down the line in a computational analysis because it might be the ontology you use to interpret the results versus something that's going to help you plan the experimental design, et cetera, right? So this quick example for reuse, which a lot of this is really driven around data management planning, sharing and reuse, honestly. So I think the better you organize your multi-omics data and the cleaner and structuring unstructured knowledge is really important. So the one cool thing that I think we've been working on is there's this one below called recommended for reuse, because we want to think about things in a way where we're not always just depending on what a database and a journal tells us what to publish. You know, you're the experts. You know exactly what it takes to reproduce or reuse or integrate that. So when people talk about machine learning, AI, and knowledge extraction, there's a lot of challenges there. You can't extract certain information from tables and figures. You're not going to find it in a supplementary if it's not well, retrievable. So one thing we have here is recommended for use by domain experts. So it may not be required by a database or journal data sharing publication policy, but it's recommended by domain experts to further support the transparency and reuse. And right now, these are things that are not really well structured, but I think if the community is giving feedback to those service providers trying to support them, then it's a good way to think about Could we do better? Is this a status quo? What would be our wish list to see? So this kind of gives some flexibility in how we're down selecting and looking at different standards. So here's something that is very well in the hard for the works, and it's something to think about because, we haven't defined it. I think we were going to build the landscape, filter down, try to think about FAIR in a different way, and then reduce it into our collection at fair sharing. But it was a lot harder than maybe we thought it would be. So this is kind of in limbo. I think right now the demo will show you for the collection is we pushed everything in from our landscape and we may filter down. There are others in the community working on fair metrics and also indicators, and that's not our space to develop here, but it's a great opportunity to adopt it and apply it. So for example, this is what's already out there, and we're using some terminology just for fun from, some other efforts that can be reused. So in terms of findability, when we are thinking about standards for our data, usually we get these persistent identifiers in a database, right? In geo, massive, wherever you put it. But there's another element to it. So is there, to incorporate the FAIR standard curations, right? So is there a mechanism by which you can communicate these protocols? So one universal one that we have in our tab is, and this is just one of many, is identifiers.org. Can you consistently provide a mechanism to share that accession and redirect to a location within your reporting guidelines? So these are kind of centered around the different standards that we're curating. And this would just be an example. If you're familiar with this kind of data, this might make sense. If you're not, so this is kind of on the back burner. We're really going to try to work with those in the community who are establishing these and see if we can be a part of a use case to support those efforts. So to our MOMSI collection. So this is the next stage, our deliverable 2 coming from 1A. So this is a demo release. Special thank you to Allison Lister and the governance team at Fair Sharing. They helped populate and export most, if not all, of those in our landscape Google Sheet into the collection. So right now we have 182 linked standard across the different domains. And then they also will link into different databases, and there's also a couple collections. And so the question for us is, why did we choose Fair Sharing as a way to build a collection from our curation review? And that's because it's an ongoing effort that has machine actionability, and it's a place where no matter where you are, if you're a postdoc in China or somewhere, in the global South, or maybe in California, it doesn't matter. You can access the exact same collection of standards that were curated by experts. So, why should researchers care about making their multi-omics data fair? And how can we best assist them prior to integration? Because there are many challenges way before integration that exist. So there's another element to this. So the bonus about using these collections is you can highlight the different relationships between these standards, right? If one depreciates another, if one recommends another, shares code or data, implements, extends. And so these are the different types of relationships and the different types of records in Fair Sharing. So you'll see this big swarm, but if you zoom in, you'll see a lot of very familiar standards, maybe reporting guidelines across different omics that are using the same ontology. Maybe they're accepting similar formats. And so this is what we'll set up for the next gap analysis, which I think some of our speakers have really great examples on coming up, and you'll kind of see this is, once we know what all these standards are, how do we identify points of integration? So if in one domain, they have less standards around sample experimental collection, maybe, and another one does, is that something that they can adopt and work together with in terms of how you build a multi-omics platform? So this is something that's really exciting. And there's a couple other features in Fair Sharing that we'll kind of go over next in our working group meeting. So if you're interested to see what types of analysis and reports come from this, very, very possible. So please join the group. And then let me see if back. I have a couple minutes just for, I'll be very respectful of time. Actually, we have quite a bit for a little bit. If anyone has questions or specifically about either the landscape review or the collection or the standards. Which one? Keep going. Here? Oh, did I pass it? Oh. Here? Okay. Oh, sorry. Thank you.
So I see there are 100 and something standard, Linux standards, which is a lot. So do you endorse any of them?
So most of these standards are endorsed within the domain expert fields already, right? So by experts in the field of genomics or proteomics, these are well-defined and accepted. It's a well-known community standard. So those who are in the proteomics field, the standards that apply to that, no. So each one of these records in fair sharing, they're tagged also. So if you wanted to go to this collection and search only out of those 182 what applies to genomics, you can filter that list and also download it. And you can add as many different search features as you want. But these right now are what exist in the community. I think there's a few that have been existing for 50 years maybe and could use a reevaluation. Do we even remember where it came from? Was it ever published? Or we just kind of kept using it for some reason? So there's, for the most part though, they are domain community accepted. So I think we heard earlier in the talk today, I think Danny even touched on it, is working with experts to understand what standards actually apply to new services and applications. And so this list is just that. And so that's kind of what our experts were helping us with. Does that help?
Yes and no, because I still think it's just such a big number and there are not that many data types. It seems that there are multiple standards per data types from just looking at the numbers.
Well, yes.
Maybe I'm wrong.
Yes.
You're right. Even if the omics, it might be 3. I don't know if you want to count genomics, proteomics, metabolomics, but within metabolomics, there are different standards. Exactly. So, but there are the ontology, there are the standard, there are the ontology for actually, yes, there are, that's exactly the point. There are a lot. Yeah.
And let's not forget the life cycle of the, for each step along the whole experiment and study, you use different ontologies. If you're preparing a sample, you use the sample ontology, and relevant to say metabolomics.
Thank you. Yeah. Is it working? Okay, right, yeah. So my name is Svein Gundersen from Elixir Norway. I have one particular question in terms of specifying models and formats. So that is one if I understand correctly, this is a category that came from fair sharing, right? Such, because to me that's very different if you are specifying metadata or the actual data. And is that the difference something that you have been dividing? I mean, is it indicated which of these models within the model format are specifying metadata and which are specifying data?
Yeah, I think. Yeah, no, that is a good question. So I think in the collection, it kind of clarifies it a bit, depending if you're looking at a domain specific model or format. So in our landscape review, it'll give you the definition of what they are. So it could be a data file format, or it could be a model schema, right? So model schemas typically will contain a lot more metadata. It may even have other standards embedded in that structure. And so the thing that's nice about that actually is if you were to zoom in, this is the really great part about having this knowledge relation graph is those will be very evident even visually in the graph itself from our collection. So these the different standards, if you zoom into them, you'll see they're the green little nodes. Those are all standards, and that's of the 4. So if you identify one in here, that's, I can't really say that well, but we have some general ones where there are model schemas, and you'll see those connected strings to other standard types like the terminologies or things like that. So does that help a little bit? Yeah. If you are, you're already part of the working group. Yeah, just if you go to it's like there's the example for genomics. We have the blue is the model formats. So if you look across them, you will actually see file type formats or other types of formats or models. So not to be confused with like causal modeling or things like that, right? Like things that are a little bit more computationally specific.
And basically, yeah, looking at the clock very quickly, some of the data, it's important to make that distinction, because in some cases, if you're describing an experiment, your metadata obviously just, for example, type of sample, blood, lung, or tissue, etc., right? Wherever you're taking it. But then when you're analyzing the data itself, for example, for metabolomics, you have the actual identifier, INCHI codes or SMILES, or the peaks, and that's the data itself. And so that, are there standards there? And so that's an important question.
Yeah. Do you have another question?
Yeah, we're doing the same. It's just a comment on the same question, if I may. Yeah, but the reason I'm asking that also because I'm co-chairing another working group in RDA, which is verification of genomic annotations, and there we very clearly separate that we are just interested in the metadata standards and not the data, because that's what we're focusing on. So that would be nice sort of just filter on that in your collection, just as a use case. Yeah.
Yeah, I mean, most of the standards that we're aggregating and curating is specific to leading into metadata, right? Metadata is really the high currency in interpretation. So I will actually say that there are quite a couple groups outputs that we have in our universal tab. Like we have the PIDs for instruments, that is huge. Instrumentation and metadata around what instrument is selected allows you to compare apples to apples, not apples to oranges. So that's not something that's very obvious at the surface. And I think that we're also coordinating with your working group as well to make sure we capture whatever developments your group is doing as well. So yeah, I wouldn't say, I mean, the repositories deal with the raw data, but they contain standards as well, right? And those are the types of things we're interested in. Can we advance upon what exists, make things more machine actionable? How much unstructured text is in our standards right now? Could we advance it? Things like that. Is there any questions online? We're good. Does anyone else have anything to add, Tim or Allison, for those who've been, part of this at all? Don't feel obligated.
No, Lindsay, thank you. That was a very, very good presentation.
Thank you. I was just going to add to that. I think it was a fantastic presentation. Thank you, Lindsay. It's great to see all this hard work that you guys have made on the spreadsheet up in the meeting.
So just to cap off, just before we hand off to the other speakers, I think the one thing why this upfront work was done, I like this visually a little bit better, so we'll just leave it there, is we hope that this collection can provide a resource for domain researchers, but also service providers building new platforms or want to build new things where you're integrating data, that it is domain context heavy. And so, do we want to develop things that have a shelf life of 2 years because we didn't have conversations with the domain experts? And I think domain experts would love to make their life easier by having tools and support services that support what we do manually right now, because there's still a lot of manual work. That was part of my motivation sometimes. But also, with our maintenance planning, I'll get that in the closing remarks, which will connect our deliverables and how we plan to sustain these going forward. But really just shedding light on the work that's been done. No more reinventing the wheels, no more new standards if they're not necessary. There's been a ton of work done in the domain community. And so I think there's not been a lot of discussion about that lately. There's been a lot of how do either repositories or services support something. I think it's a matter of building knowledge around standards and then building technology into it. So with that, I will cue this up to our first speaker, Tim, and co-chair. And this will be these are really great talks from all three about how this applies in the actual field.
All right, thank you very much.
Can you try sharing, Tim?
Yes. There we go. Can you see something?
Yeah, now yes. Thank you.
All right, great. So I can't see the chat. So if there's a question or there's something wrong, please interrupt by saying something. Otherwise, I won't hear it. All right, but yeah. First of all, hi everyone. I'm Tim van de Bossen from the Compomics group in Ghent, Belgium. And first, I want to thank RDA for having me in San Jose, and I really want to make them person. But for familiar reasons, I had to cancel last minute. So hence why I'm doing this online. Nevertheless, I'm very excited for this talk. So today I will talk about how I'm currently squeezing metagenomics metadata terms into proteomics. So I'll talk about public data sharing efforts in proteomics and metaproteomics, the metadata efforts in proteomics and metagenomics, and what we can do next. So for over a decade, the Proteome Exchange Consortium has brought the most proteomics repositories together under the FAIR principles. And they have been highly successful in this effort, with mandatory submissions from journals and funding agencies playing a key role here. So indeed, and as we see over here, there's an increasing number of datasets submitted to Proteome Exchange. Now, in metaproteomics alone, we have 390 datasets. So the number of metaproteomics publications is just for reference here, totaling around 1,300, with the first one in 2004. By the way, these numbers are from July, so approximately during the half of the year. But it's interesting to see that after the start of Bright repository in 2005, which was the first one, it took 9 years to see the first metaproteomic dataset appearing. Now, importantly, this is about the data and not about the metadata, because that's a whole different story. Now, mostly when I start talking about metadata, I receive the same message. Oh, but Tim, it's in a manuscript or it's in a supplementary, but I do not agree with that. So Tina Kleist from our group, Compomics, she led an effort to manually annotate over 240 Pride projects. And this is the missingness of the metadata. In orange, you can see the missingness in Pride, and in red, the missingness in the publication. So that's publication itself, the supplementary information, or basically anything we could find. This just means that a lot of data in Pride is not reusable and not reproducible. And yes, that's a big issue. But it's not the new issue. So it's not that we have just discovered this, or it's like a new finding. Now, in 2021, the HUPO Proteomics Standards Initiative and the UBIC-MS initiative developed SDRF Proteomics, a metadata standard which is used in transcriptomics as well. So what does SDRF Proteomics do? Well, it takes the properties of your samples, like what tissue they came from which species, what you're studying, and that combines that with the properties of your data file. That includes the type of instrument you use, the fragmentation method, the modifications, and so on. And crucially, the last part is also the study variables. So, for example, whether the sample came from a healthy donor or from a sick donor. And SDRF Proteomics basically ties all this information together. So, is SDRF Proteomics easy to use? Well, first you go through the manuscript to grasp the content. Then you have to visit the GitHub page to find examples, templates, and detailed information on the structure. After that, you need to navigate through about 14 different ontologies to find the terms that fit your research. Then you have to create a flowchart of your research and finally combine all of this information into a spreadsheet-like software, like Excel, for example. Well, it's not very easy to use. And if you want people to use SDRF Proteomics, it should be easy to use. But let's have a check. Like, do they use it actually? So if you currently have a look at Bright, There are more than 27,000 datasets. It's the biggest repository we have. So more than half of those datasets were submitted after 2020 when SDREF was developed and published. So you would assume that most of them have SDREF annotation. But no, only 200 projects upon submission from the original researchers annotated their data in a machine-readable, reproducible format. Now, in parallel to this, a lot of people have been annotating data post-submission. So they're not the original researchers, but they annotated also 230 datasets. That's after submission. That means that less than 2% of Bright is annotated. And that is a real issue, especially since we're moving to a data-driven era where we want to reuse public data. That's just not good. So my colleague Klaes, she developed LessDRF, which is a Streamlit app to facilitate filling in SDRF files. So I don't have to get the time to go through this, but it's very easy to use. It's a Streamlit app. So if you scan this QR code or go to lessdrf.streamlit.app, you can test it yourself. So even though it's fairly easy to use now, and it's very easy to fill in an SDRF file, there's a big elephant in the room. It's not an elephant, but it's a bacteria actually. So when performing a metaprotein experiment, it's just impossible to capture all necessary metadata in current SDRF files. And therefore, we need to have more than one different file for metaproteomics, and even more, one format per environment that we study. So the only thing left for us to decide is which additional features to add per environment. Looks easy, but turns out that different researchers and experts have different opinions about it. Who knew, right? So luckily, with the efforts at GSC, we might be able to solve this. So in GSC, they have developed environmental packages and extensions, which we will try to implement in SDRF proteomics. So how are we going to do that? So we'll take a default SDRF proteomics file and we'll merge it with the environmental packages or extensions from GSC. Note that Mixes itself contains the technical information about the experiment, so that's not very useful. However, the extensions and environmental packages, they provide the information about the environments. And yeah, that's the part we need. So, and more specifically, we're going to take those attributes and make them SDRF proteomics proof, so it will pass our SDRF validator. So what's next? We have the concept already. What do we have to do? So first, we need to get the mixed ontology or terms in SDRF proteomics. That's basically, that's currently ongoing. So with the help from GSC and the people from Bright. We should also ensure that the characteristics organism fields should be either removed with SDRF metaproteomics or be made optional because, well, metaproteomics, we study bacterial environments, right? But the environment itself should be added. Additionally, we will also add a biosamples field, so which is useful because we would like to work towards multi-omics studies, which perfectly aligns, by the way, with what WOMSI is doing. And then finally, we need to get this approved by the scientific committee of the Metaproteomics Initiative and also approved by HUPO-PSI, so the standards initiative from the proteomics community. And afterwards, we will also implement these templates into LessDRF, so it's very easy to use for researchers to use these metadata templates. Now, it's also very important to realize that this is just not me wanting to introduce metadata in the field. No, that is actually also backed by the Metaproteomics Initiative, whose goal is to promote the dissemination of metaproteomics fundamentals, advancements, and applications through collaborative working in microbiome research. And this is the standardization part, is one of the three pillars where the initiative is, well, what the initiative supports, next to the collaboration and communication pillar and the education pillar. So By having said that, I want to thank you for your attention. But I also have a call to action. It's basically, I need your advice. So what should we take into account? Are there any mistakes or bad practices we could avoid? Do you have any recommendations? All feedback here is more than appreciated. So thank you very much. These are my colleagues. So thank you very much.
Try to hold all conversations or questions until the end, so we get through each speaker, and then it can be kind of a free for all for a little bit. So let's move on to our next speaker.
So Bert, if you can try share, perfect. Okay,
Yes, can you hear me?
perfectly, thank you.
Okay. First of all, thanks a lot for having me and also for letting me meet Tim, because we are actually part of the same building. But before this, getting involved in this event, I was not aware of what he was doing. So thanks a lot for that. I will be talking about MARS, Multi-Omics Adapter for Repository Submissions. And as you can see, the acronym was first, and then we came up with an explanation for it. I forgot to mention, I'm part of the Flemish Institute of Biotechnology, the Data Core group, and also part of the Elixir Belgium group. And yes. So first, a brief introduction. Within Europe, we have EBI, who actually created multiple repositories for every omics field. And this allows researchers and bioinformaticians to submit their data towards these repositories. And for the genomics field, we have the European Nucleotide Archive, ENA, and also the European Genome Archive, EGA. For transcriptomics, we have ArrayExpress, for proteomics, Pride, metabolomics, Metabolites, and for metagenomics and metatranscriptomics, Magnify. This is very nice for publishing the data, but currently all these repositories have very different ways of submission processes. So not only the way how the data itself needs to be submitted, but also the metadata, due to different standards that are in use, is quite different. And another part is that if you would do these submissions, or treat them all separately towards these repositories, you would basically either submit multiple assays that are done on the same samples. You would submit the sample information multiple times, and you might also lose track of the scope of your multi-omics study. Luckily, EBI has for this the biosamples, where you can either register in advance your sample inside of biosamples, you get an accession number in return, and then this accession number, this registered sample, you can reuse then throughout all your executive submissions, or you create new samples and biosamples, which you then link back to the original biosample identifier if, for example, the repository would not support the linkage towards an existing sample in biosamples. And yeah, so this actually allows the researcher to keep track of the original source and then also the relationships between those data sets. And so these two concepts is the thing that we within the Mars project try to simplify and improve. And for this, we actually want to make use of the ISA JSON format. I will talk later more about this. Within this initiative, I think one of the unique selling points is that we work closely together with the repositories themselves at EBI, and with then data infrastructure professionals who are hosting data brokering platforms or other platforms that create as adjacent. And the idea is that Mars will help out in doing all these submissions and also make a difference into helping out repositories and how they can accept metadata in a more standardized shape. The more technical parts, but also more information on the project itself can be found on the GitHub of Elixir Europe under the Mars repository. And yeah, you also can here use the QR code to find more about this. This diagram gives an overview of our project, where I will introduce the three main stakeholders. So we have the platform that produces the ISA-JSON, which feeds the ISA-JSON towards the Mars CLI, which is hosted by the data brokering platform. And this Mars CLI then will broker the metadata and data towards the repositories on the right. And all the communication of metadata is done through ISA-JSON. So we have three stakeholders, of which the first one was the ISA-JSON producing platform. And it's really at this stage, at the very beginning, when you are describing the metadata, that we will do the enforcement of using the correct metadata standards, and also that actually the necessary fields are being used. So we can almost at this stage, or we try to guarantee a successful submission, and this by enforcing controlled vocabularies, ontologies, and all these kind of things. The current ISA-JSON platforms that we have in mind are Arc, Seek, the ISA Wizards, and the ISA Creator. Then in the middle, we had the data brokering platform, which can be Galaxy or your local computer, or a cluster, or other custom tooling. And this platform will take care of the authentication. And then we're talking about also the credentials of the repositories themselves. It will also make sure that our Mars CLI tool has the necessary data files for submission, that the ISA-JSON is there, and it will also then host the Mars CLI itself. And then as third and last, we have the EBI repositories themselves, and they will maintain an API endpoint that supports ISA-JSON. And I think the important part here is that it is maintained by themselves, and they will also work closely together with us, so that we know that when they make changes, and also the opposite around, when we, for example, make changes in how the ISA-JSON needs to be interpreted, that they are aware and then can adapt their adapters accordingly. These, I will touch on that later. These repository endpoints will also return a standardized receipt. So most of you probably are familiar with ISA-JSON, but if not, I will repeat it here. So the ISA-JSON is a well-known standard within life sciences, and is built on the investigation study assay principle. And it is really designed for the kind of thing we want to achieve here. So multi-model studies across various domains. And Particularly the study and the essay is important to us to store the metadata relevant for the repositories. And then also when there was a successful submission, that the returned accessions and identifiers can be stored back into the ISA JSON with an example on the right. Really at the core of the project, and also the more tangible thing that you will find in our repository, is the Python library or command line tool. You can use it in both ways. Will take care of the ISA JSON ingestion and also the syntactic validation, guaranteeing that it's a valid ISA JSON and that it also contains the necessary attributes for us to figure out to which repository one wants to submit. It will also then take care of the actual submission towards these repository endpoints, and that the receipts that are being returned are being parsed, and that then also things like error handling, but also asynchronous validation steps are taken care of, and that then the returned accessions are stored in the ISA JSON, and an updated ISA JSON is being dumped. And also, of course, I forgot to mention the data submission itself. So on the repository side, they will deploy and maintain an ISA JSON endpoint, and we there work together in order to figure out how they have to fetch the necessary metadata inside of the ISA JSON, because people who are familiar with this standard know that there are many ways of achieving similar things. So this is crucial. And also the standard that this endpoint will return this receipt is standardized within our project. Yeah, that's about it for this slide. This is a slightly more complete overview of the project, highlighting the interactions between the stakeholders and the use of ISA JSON and the standardized receipt. And then also here, the data brokering platform that is handling also the data submission part using the Mars CLI. Currently, we have a proof of concept working of everything that I've said, using by submitting towards ENA and metabolites using an ISA JSON, and by first registering the samples inside of biosamples and then reusing these biosamples accessions throughout the executive submissions, and then updating the ISA JSON and returning it. Currently, we are also working with the ArcSeq ISA and the ISA Creator ISA JSON producing platforms to try to support from early on a wide variety of ISA JSONs. Towards the future, we would like to onboard more repositories, and since we've done quite an effort recently into describing how repositories should use the ISA JSON, this should go better and faster. And for this, we are already closely collaborating with the EVA, the European Variant Archive. And we also initiated conversations with Magnify, and also Pride is on our radar. When it comes to the data brokering platforms, we would really like to make a Galaxy tool for the Mars CLI. So yeah, Galaxy is a web platform to do data analysis and workflow management. And having the Mars CLI there would mean that researchers can not only perform analysis on their data, but then also do a submission from the platform itself. And we're also bringing on the idea of having a custom web interface, but this is still to be seen. I would like to thank Elixir Europe and especially the Bi-Hackathon of Elixir Europe, because this project would not have been there without it. It's already for 3 consecutive years that we were blessed with a project and that we could work on this. And last week we had the last Bi-Hackathon, and we made a lot of progress in this project. This project is also part of work package 2 of the Elixir data platform. And besides that, I want to thank you for listening.
Thank you very much, Bert. So hold your questions, but think about them till the end. So next up is Catherine. So Catherine, could you share your screen? Perfect. Take it away. Thank you.
Great, thank you. I'm Catherine Heel. I'm from the Pacific Northwest National Laboratory, and I'm here today to talk about some work that we've done as part of the National Microbiome Data Collaborative. So I'll tell you a little bit about the NMDC and then a little case study about how modeling mass spectrometry metadata in the NMDC, kind of the before, the challenges, the after. If I can figure out how to switch my slides. There we go. Okay, so the NMDC, so the National Microbiome Data Collaborative, we have 3 main uses. We have a submission portal where people are aiming to contribute and share their study and biosample data. We have a portal for processing data with standardized workflows. And then we have a data portal where you can go and search for processed and raw data. And all of this is to support researchers to create, use, and reuse microbiome data, and specifically microbiome omics data. We have a focus on data standards and infrastructure and community building. And I think that's all really in alignment with everything we've heard from this session. So these 3 main products that we have as part of the NMDC, the submission portal, the processing of the data, and the data portal, are all intimately connected with the NMDC metadata database. And so the main role of NMDC, one of the main roles of NMDC is to standardize, collect, and make transparent the metadata about the microbiome omics data. We don't actually house the raw data. We point to where the data are housed, so they're accessible, but we do house metadata. And so the metadata database, again, it houses the metadata, and it houses metadata from all of these different steps, from how the samples were collected in the environment, how the samples were processed in the lab, how those lab samples were then sequenced or used on a mass spectrometer to create raw data, and then how those raw data were processed into processed data and digestible data. So we have a schema that represents each step of this process and also the material entities between each step. So we have a schema that captures kind of what this process data, what this process sample, how it's connected to each step along the way. And as you can imagine, that's pretty complicated, and it is a work in progress. And so initially this metadata schema was designed by and for biologists, and specifically folks with a lot of experience in metagenomics and metatranscriptomics. And so once folks like me, who have more of a background in metabolomics and proteomics, mass spectrometry side of things, we realize that this step of what we're calling data generation, so going from a sample in a lab to a file on a computer, so either sequencing or mass spectrometry, which Lindsay really nicely laid out, that this modeling had to be expanded in order to really make this aspect of the metadata conform with standards and to make our metadata fair in this regard. So I'm going to show you before what this aspect of the schema looked like. So we had the records had to follow this particular format. They were called data generation. They had an input, which was a physical biosample or a sample from a lab. It had an output, which is a data object. So these are the data objects that we would then point to, say, metabolites or a different NCBI or some other data repository. The instrument name was the type of instrument that was used. The omics type, which was a constrained value like metaproteomics or genomics, transcriptomics. The process and the processing institution, another constrained value. So these fields are applicable to both sequence-based and mass spectrometry-based applications. So that's great. We also had some fields that were only applicable for sequence-based applications. Some fields having to do that were capturing PCR conditions and nucleic acid amplifications, things that as a mass spectrometrist, I would never need to capture on for my metadata about how the data were generated. And then several fields were flat out missing that are really important for capturing for mass spectrometry-based applications. So now I'm kind of moving to the after side. This is how we've remodeled this aspect of the metadata so we can capture more complete information. The first thing we did is we subclassed. And so now all of our data generation are either that of nucleotide sequencing or mass spectrometry. And this also enables us to expand if we want to expand further. So we could start incorporating NMR-based data generation, which would also have a different set of metadata that we would need to capture. And so that subclassing really opened up the door about how we can model specifically mass spectrometry information. So we've kept the fields that were important for the sequence-based and mass spectrometry. So I'm not going to kind of re-go through those. We've improved some of them, but we've kept them. And then we've also added fields that were specific to metabolomics and lipidomics using the standards as best as we could that we've already been talking about today. And so this was in part just explaining very explicitly how are these samples introduced to the mass spectrometer, like gas chromatography, liquid chromatography, or direct infusion. And then really importantly, we've now linked to two other records that capture the configuration of the chromatography, if applicable, and the mass spectrometry. And those are, we can think about it as modeling the literal methods that ran the instrument. As far as the liquid chromatography, that's my kind of specialty, and the mass spectrometry. So you can think about these are the metadata about the methods that generated these data in mass spectrometry. And so now we have an input of a single sample, an output of a data object, but many of these data generation records can hold, can share a single method, and that's really important. And so I just have this kind of before and after as a kind of example of the things that really not much changed. The subclassing was huge. These fields are no longer the PCR nucleotide sequencing. Those are on that subclass, but not on the mass spectrometry subclass. And this was a big unblocker for other aspects of NMDC. And so I really just want to highlight how this restructuring of the metadata, the restructuring of the schema in order to hold more applicable metadata really unlocked some things. And so the first thing that it unlocked is that we can improve our mass spectrometry-based workflows that we also have part of NMDC. So now the workflows are configured. So we are telling the workflows how to run based on the configurations of the data generation. So everyone who's done any metabolomics workflows knows that you have to treat GC data very differently than LC data. Now that we've captured that information in the metadata, the workflow can go and look at the metadata and say, actually, we need to use this workflow instead of that workflow. The second thing that's really important for lipidomics, metabolomics, and proteomics is now that we can say that all of these data were generated using the same method and in the same batch, we can start to do relative abundance between samples. Before, we were only doing one sample to one observation, essentially, and now we can start to connect those samples between if they're run at the same time in the similar manner. And finally, this really is going to enhance searchability on the data portal. So this is, again, a user need is if a data portal user wants to find all the data in this kind of environment that was collected with this type of instrumentation or this type of data generation. Now, by exposing those configurations on the data portal and say, this is GCMS, this is LCMS, this is DDA, this is DIA, that kind of information can be exposed on the portal and made really accessible to users. And so I like this as a case study because the metadata is really the foundation for the products that we are producing. And the mass spectrometry omics data have very different metadata, especially concerning data generation, than the sequencing-based omics data. And it wasn't until we were sitting in the same room of the mass spectrometrists and the data modelers that we were really able to communicate and explain why we needed this different type of information. And those use cases of like, here's what users ask for, here's what users need, and then evaluate our metadata schema, massage it a little bit, change it, and update our metadata to enable that. And so that's all I have. And here's the project team, and the funding is through the Department of Energy and BER in particular. Yeah, thank you.
Just want to say thank you to our speakers, Tim, Bert, and Catherine. They all did a really great job of explaining where some community developments exist. I think a majority, if not all, of the standards mentioned today are in our collection. So you can explore those and the relationships with those. Right now, we have a couple of moments if anyone has any specific questions for any one of the speakers, online or in person.
I have a few questions. I guess I can start with Catherine. So it seems that your users need to do two submissions, right? One for your portal and another for the actual data housing repository. So it's double work. How do you enforce that or encourage?
Yeah, that's an ongoing challenge for sure. So the submission portal right now is for study to biosample information. And right now we're mostly retrospective. So we're trying to connect existing data in repositories to metadata. And so right now we kind of take care of that connection as far as if it's already in a data repository, we'll connect it to our metadata repository. And we have some ideas, and I think they're evolving as far as how we can bridge that for people who are starting with data in hand and they want to do it all at once, or backwards. If they've already submitted it to a repository, how much can we glean from there to bring it into NMDC, and then kind of say, can you help us elaborate on these points if that repository doesn't require all the metadata that we think is required in order for these data to be fair and reusable. Does that answer your question?
Yeah. So you do, right now you do most of the work and just trust your users to fill the blanks, I guess, right?
Yeah, that's kind of the scenario we're in right now. And I will admit that part of the metadata I'm not as familiar with, but I know that it is definitely a challenge and ongoing.
Yeah, one would think that the most straightforward way is to convince the raw data repository to house all the metadata, right? But I suppose they don't have much flexibility and or desire to do that.
Yeah, and I think one challenge is the raw data repositories aren't, NMDC is specifically focused on having, reusable data for environmental microbiomes. And that requires, like we've learned, specific metadata that a repository that's just that's housing metabolite data, regardless of where it's coming from, asking them to say, require these certain types of metadata is a big ask.
Mm-hmm. So environmental being not from parts of human body, right?
Or not, yeah, not clinical in particular.
Okay. So, well, Mars to Earth, are there any plans to expand this tool to US-based repositories?
It looks
Bert,
like
are you
Bert is
still-
it looks like Bert is offline.
Ah,
Or
okay.
he's
All right.
yeah.
Maybe his connection got-
It's indeed an open collaborative initiative. So if there is an interest in doing it and somebody is willing to join to make, because it's just a matter of writing codes, then I would say yes. Well, it's it's an unfunded collaborative thing that happen under Elixir just because we targeted obviously EBI, which is a node in Elixir repository. But absolutely, this doesn't exclude that any other repository could be added. We just need somebody they're willing to volunteer time and coding skill to just get connecting, get involved, get familiar with the format. The other will help, and then write the transformer that's needed for the repository. Yes.
So another question is, from what I saw on the schemes, you start from the samples. So it's maybe organism and tissue, but do you collect any information about that came before that? So exposure, disease, treatment, demographic.
So because I'm familiar with the ISA format, I can answer. The answer is yes. ISA, it's investigation study assay. So it has enough flexible metadata for you to collect any contextual information you need. Yeah.
I had some technical problems, so
Oh,
sorry if I
Bart,
missed
you're
question.
back.
Yes,
Maybe
I had
you
to
can
restart
answer
my
in detail.
computer.
Right. I tried to answer to, so there was a previous question where the question was, why not extending it to repository in the US? And I said, if somebody is willing to join the effort and put a bit of time and coding, I see you are nodding. So that's, I answered that, but there was a second question. So I don't know if you heard.
No, I actually did not hear any question towards me, but what you say is it's for sure true. And currently it's focused on EBI repositories just because we have this good collaboration within Elixir with EBI. But of course, towards the future, towards American or Japanese repositories, it's for sure not a limitation on the technology and our concept. It's yeah, you can also see this presentation as a call out for more repositories to onboard. And the
So
other question.
Yeah, my second question was about the sample information that is more clinically relevant, which is information about exposure, personal exposure, disease stage, maybe treatment, maybe health outcomes or demography. So, and sorry, I don't remember your name. The lady here, Susanna.
Bert.
Susanna said
Susanna,
that
yeah.
the adapter has enough flexibility to accept this information because of the structure of the ISA standard. But where will it live? Because I don't believe that repository themselves have place for this metadata.
Yeah, so this is something that we also discussed quite extensively in the sense that depending on the repository, sometimes you can store more metadata than the repository itself displays or exposes towards the users. So in any case, then indeed it can be accepted by the adapters and submitted. But in general, when it comes to like publishing the data, you're always a little bit limited to what the repositories themselves index. The ISA JSON itself, of course, is very there is no limitation there in what you can store, and we see that we store the metadata per repository. So you have for every repository an assay in the ISA JSON where we will store the metadata, and there we can easily store more metadata than is required, more protocols and these kind of things, and especially the things you mentioned. And these things will be preserved then in our ISA JSON and inside the metadata platform that will produce this ISA JSON, but it might indeed not be visible in the end repository, depending on what they index there. I don't know if this answers your question.
Can you search this adapter? So if you store the metadata that is in this intermediate, can you search it? So maybe this metadata is not going into the target repository, but will you be able to look for that in the?
This on the repository side, this depends from repository to repository, and
but
on
the intermediate, you just said that this intermediate does store all the metadata that the user entered. So is this intermediate searchable?
Yes, but you're searchable from depending on the platform. So for example, if you would use Seek to create your ISA JSONs, then there it will be searchable within your project where this metadata will live. And depending on how you share it there towards the outside world or internally within your institute, you will be able to search these things. But there, the thing is that we provide mainly the middleware and how to use the standards, but the capabilities you're now touching on really depend on the ISA JSON producing platform and the repositories themselves. So yeah, it's a little bit out of our hands.
Hello, my name is Ricardo. I'm from Costa Rica, and we have I'm from part of the staff, but I'm from part to the other part of academic and research. I'm researcher of the National University of Costa Rica, and my question is about Latin America. And I am interested. I'm really I don't know how to say that, but let me see. Okay. Well, congrats about all the efforts and all the presentation. It's really nice, and we have to learn about that. We have to learn about all the process. But my question is about how to make a culture of data and of to be more open access right now here in Latin America, because we have a lot of problems with these situations, because we have a lot of research that is closed, is not open, and it seems that it's difficult to open the mind, and it's a open question, of course, but I have to learn about that, and we have to learn about that. Yes, thank you.
That's why we're here.
Yes, thank you so much.
Thank you for hosting us.
Oh, we have a question on, or go ahead, Tim.
Well, it was more as a reply to the question that was posed. Yeah, that's a very good question, actually. It's a question that basically comes up at every data sharing or metadata sharing event. And for me, it's always about well, is it the carrot or the stick, right? So do you incentivize people to share their data, or do you punish them when they do not share it? So what we see in proteomics is that the journals and the funders require us to share our data publicly. I'm talking about non-clinical proteomics data, just in general, and where it's not where you cannot trace back the sample to the person. That's an ongoing discussion we're having right now. But yeah, so if your data is just not deposited in Pride or any other proteome exchange repository, your paper will not be published. It's as simple as that. And I do think we should do the same for metadata, because we have now a tool called LessDRF. I talked about that, which makes it very easy to annotate your metadata in a format which has been agreed by the whole proteomics community. We should try to enforce the journals and the funders to ask the same as what they did for the data deposition. Now everybody is depositing their data. They don't even think about it anymore because they're used to it. And we should create the same culture for metadata. But that's how I see it.
So I think just a couple points. I thank you all for being patient. Just a couple more minutes. I think what we're hearing in this session today is that there's somewhat of a paradigm shift in how we're sharing our data and metadata and what it really takes to move forward in a automated and technology heavy world. So I think hopefully these collections and the things that we're doing open the conversation. I think what we've seen between the three speakers is that there are open conversations happening between experts, stakeholders, and developers. And there's flexibility and there's openness and there's a safe space to have these discussions, right? And I think for those who might need more assistance with like open access or metadata and what's the state of the development across, globally, hopefully we can illuminate that in some of our deliverables, and that's the goal there. So I think revisiting these discussions with journals and databases to expand what we really need, and then also making it flexible for use case without reinventing wheels and doing the same thing. So I think just keeping eyes and ears open is very important. So just to touch base a little bit on some closing remarks and next steps, we do still have a couple more deliverables that we're working on. So, we just started working on the second one of the 4, but just some updates. We just got some funding from RDA Tiger. So that Google sheet that you just kind of viewed, which is not machine accessible and it's not in a format that can build sustainability moving forward. Main aim is we've done all this great work. Let's keep it going. Let's keep it inclusive. Let's keep it open. So if any of you have had a chance to walk around in the auditorium at one of the Tiger stations, they had the maintenance platform dashboard. So Wim Hugo had that up. We are going to be doing the same thing with our landscape dashboard. So a couple of things. We'll have a backend GitHub repository for storing and then also if people want to contribute to our collection at fair sharing, this is also a mechanism to request that, adding new records or updating, but also it gives you a way to explore maybe more information you want to know, like who are the affiliations building these standards, it kind of gives you points of contact for new discussions. So there's going to be quite a few of these deliverables, but they will be open. And then it's also hard in defining moving forward. How do you select from this large collection? What is the best way to build your own collection? Let's say if you don't want to use the Multi-Omics collection. So those are a couple of discussions that are ongoing. And so this next, what we kind of heard from speakers in our next deliverable or integration gap analysis is hoping to find some of these points of or challenges that you heard from some of our speakers. So where can we adopt standards that are missing from one domain to another? And how do we make that selection transparent and reproducible? So right now, I think there's a lot of great developments, but on paper, it's not structured. There's no process, there's no guidelines. So maybe we can help assist with that. And then lastly, in collaboration with Fair Sharing, of course, we have a collection fact sheet coming out. So this will give you a little more information about what is a collection, what can you do with it, how do you use it? So for those less familiar with the interface, and then a couple of the co-chairs, and then any members, of course, we are putting together a landscape review article. So what did we identify in our landscape review? What is the state of domain developments across the different omics? And then how do we build discussions together instead of in silos or behind closed doors? So with that, a quick special thank you also to a few members. I know Eric Deutsch and Nils Hoffman did help a lot with some of the landscape selections. They worked with Tim and I, and then of course, Fair Sharing, Alice, and everyone helping with the collection. With that said, please join our working group. There's a lot of great things to come. This is just the beginning. I hope everyone can at least take something home from today's discussion. And then with that, if there are extra questions, feel free to chime in. If not, then we appreciate your time. Thank you.